Hey everyone, welcome back. So over the last few videos, we have learned how to evaluate sequencing quality and use cut adapt to trim away bad data. Okay, so we now possess a FASTQ file full of pristine high quality DNA reads. But a single read is only like 150 letters or something long, but the human genome is 3 billion letters long. So right now our data is like having a million jigsaw puzzle pieces dumped on the floor without the picture on the box. So today we are going to solve that puzzle. We are going to take our tiny reads and figure out exactly where they belong on the massive reference genome. And this process is called sequence alignment or mapping as well. Okay, so to do this, we are going back into the terminal to use an absolute legend in bioinformatics, which is called the Burrows-Wheeler Aligner or BWA for short. So let's get started. First, let's open our VS Code terminal. If you are on Linux or WSL, you can install BWA easily using this command. First of all, let's start WSL. Once that has started, you can simply run sudo apt uh, install BWA. Okay, then enter your password. That will install BWA in your subsystem for Linux. If you have Conda, you can run this command Conda install C Bioconda and then BWA. You can use this command to install BWA if you have Conda. Okay. Next, to understand how this works, let's create a very tiny simulation. Okay, we need the picture on the box, which is our reference genome. So I will use a quick command to write a tiny fast file with just one chromosome. Okay, so what I will do is echo, and then I will take the chromosome name. Okay, and then I will save it to reference.fasta. Okay, so a reference.fasta file has been created, as you can see over here. Now I will add a sequence to this FASTA file. Okay, this will be our reference sequence. So echo, then we will take a sequence like this, then we will save it to reference.fasta. Make sure the spelling is always correct so that you don't make any mistakes. Right, so as you can see, the sequence has been added to our reference file. So now we need one of our puzzle pieces, which is a clean read. So I will create a tiny FASTQ file containing a sequence that we know belongs in that genome. Okay, so first of all, let's create that file. Okay, so let's call it a read1, and we will add it to clean reads.fastq. Okay, so as you can see, clean reads.fastq file is created and read1 is ready to be entered. Okay, so we will enter read1 over here. Let's take GATTACA. Okay, and we will save it to clean reads.fastq. Okay, that sequence is now here. Next, we will add a plus sign, and after that we will add the quality score. Okay, so let's just take I. How many times should I take it? 7 times, I guess, right? Okay, 7 times. And that quality score is now added. Okay, so now we have our reference.pasta and our clean reads.fastq. So now we are ready for aligning. Okay, so next step. If I asked you to find the word mitochondria in a 1000 page biology textbook, so how would you do it? You would not read like page 1, page 2, and all the way to page 1000, right? That takes too long. So you would go to the back of the book, you will look at the index, and it will tell you exactly what page to turn to. Right. So computers are exactly the same. BWA cannot search a 3 billion letter text file quickly. We have to tell BWA to build a mathematical index of the genome first. Okay. So this is always step one. So what we will do is we will run BWA index reference dot fasta. So hit enter. So what happened for our tiny genome, it finishes in milliseconds. For the human genome, this might take a couple of hours since it will be extremely huge, right? So, but the beauty is you only ever have to do this once per genome. So if you type ls, you will obviously see a lot of files, but you will also see that BW created a bunch of weird new files like .bwt. Where is it? I cannot find it here, over here, right? .bwt is there, .psc is there, and I believe .sa is also there. Okay, so do not delete these files. This is the textbook index that BWA will use to search at lightning fast speed. Okay, so next step. Now that the index is built, we can perform the actual alignment. So the modern algorithm inside BWA used for Illumina reads is called MEM, which is maximal effect matches. Okay, maximal effect matches. uh, MEM for short. Okay, so the command structure is very simple. The tool, the algorithm, the reference, and the reads. Okay, so the tool, okay, the algorithm, then the reference dot pasta, and the reads, which is clean reads dot fast queue. Let's wait. If you hit enter right now, BWA will do the math and just vomit the results directly onto your terminal screen. And we do not want that. We want to save the results into a file. So we will use the bash redirect symbol, which is the greater than sign, to send the output into a brand new file called an .sam file. Okay, so what we will do is we will create a new file which will save these results. So we will call it align results.sam. Okay, now hit enter and boom. So, see, you just perform sequence alignment. So what did we just create? Let's use the cat command to look at the contents of our new file. Okay, so we will use cat align results.sam. And you will see some header lines over here starting with an at the rate, okay, and then a line that looks like a chaotic spreadsheet. This is the sequence alignment map, or SAM for short, SAM format. Okay, so without getting too deep into the weeds, this file tells you everything about the jigsaw puzzle piece. It tells you the read ID, okay, then which is read 1, then what chromosome it is matched to, which is chromosome mini genome. Okay, then at most importantly, the exact mathematical coordinate where it belongs. And if you look closely at that line, you will see the number 49. Okay, that means BWA figured out that our tiny GATTSCA read matches perfectly starting at the 49th letter of our reference genome, and it solved the puzzle. Right. So, and there you have it. You have learned why we index genomes and how to use BWA MEM to align FASTQ reads, and thus generating a SAM file in the process. But however, a SAM file is written in plain text so humans can read it. Uh, so uh, so the SAM file is basically written in plain text so humans can read it. But when you align 50 million reads, that SAM file becomes hundreds of gigabytes large, and it is way too big to store or analyze efficiently. So in our next video, we will learn how to use basically a Swiss Army knife tool, which is called SAM tools, to compress our SAM files into a binary BAM file. Sort, and then we will learn how to sort it, index it, so we can finally start hunting for biological mutations. So if this video helped the jigsaw puzzle of bioinformatics click into place for you, hit the like button and make sure you are subscribed. Let me know in the comments if you prefer mapping DNA or writing Python scripts, and I will see you in the next video.